Well, hello, my name is Lorenz, and I manage the University of Minnesota soybean breeding program here. Minnesota is a state that's located in the north central part of the United States, and so therefore we breed for early maturing soybeans that fit within a relatively short growing season. Another thing about Minnesota is that we have a fair number of specialty soybean bean producers, food type bean producers who specialize in identity preserved soybeans and contracts. And so we work with a lot of those types of organizations in Minnesota and surrounding states to deliver these types of cultivars that are suitable for these types of products. So what I want to do today, just a brief outline on where I'm going. First, I want to start off by giving a brief background on what goes into a soybean cultivar development program, what it is we do, and why we do it. A little bit of background on that. And then I want to talk a little bit about some of the current traits in our breeding pipeline, and things that you possibly might be interested in that could be coming out in the near future and that continually come out of our program. And then we'll talk a little bit about some newer technologies that we're applying in our program that will enhance some potential for some future developments. And then, of course, I'll end on some final thoughts. Okay, so first off, I want to sort of go and acknowledge the where soybeans are in the 21st century. So, soybeans are predominantly used for their meal and for their oil, turned into meal mostly for monogastric livestock feed. The oil is also used, of course, in a lot of vegetable oil applications, frying applications, and it can also be turned into transportation fuel for biodiesel. Soybeans are also useful for, of course, for a long time, been used in a lot of traditional food products. And lately, there's a growing interest in plant-based proteins in these plant-based meats, an example of which is the Impossible Burger that is offered at Burger King, made by the company called Impossible Meats. And soybean is, in this particular case, soybean makes up most of that product, and soybean has a terrific amino acid profile, making it a good source of protein for these products. So that's an exciting area going forward. So soybeans have a lot of uses in this day and age. We also expect a lot out of new soybean cultivars compared to what our ancestors who may have been growing earlier cultivars or land races would have expected. These days in the 21st century, we expect soybeans to stand up perfectly straight at harvest. We expect soybeans to be resistant, to have genetic resistance to various pests and diseases. And of course, we expect them to be broadly adapted to and have stable performance across many years and locations. And we expect them to have, good protein and oil content. So we expect a lot out of soybeans in the 21st century relative to what some of our ancestors who grew soybeans would have expected out of soybeans. And of course, producers expect high yield. Soybeans yield much more than they used to, and that is an expectation of producers. So what about that high yield? So soybean breeding has successfully increased yield by quite a lot over the years. If you look at this, one of the more recent studies on this topic, where they took many different soybean cultivars bred in different decades, ranging from the 1920s to the mid 2000s, you can see that there's been about a twofold increase in yield over these years. This has again been accomplished through the gradual improvement of yield through soybean breeding. So let's just talk a little bit about what goes on in a plant breeding program. And a plant breeding program is really quite simple. It basically boils down to three different steps. So first of all, we have genetic variation. We have to identify genetic variation or create genetic variation. And we can do that either by creating a mutation population, which I'll talk about a little bit later in the presentation. We can identify an existing genetically heterogeneous population out there and make selections within that. Or most commonly, what plant breeders do is they make a cross between two individuals, and the population of progenies resulting from that crossbreeding activity will be, again, genetically heterogeneous, which can be used for making selections and driving new cultivars. So we make our population, we create our genetic variation, and then we, of course, test it. So we have our traits that we want to test for, we have our targets, our target markets, our targets for our cultivar. We test our individuals, or we create families, or we test our families. We test them, and then we simply select the best. And we select the best by when we select the best, we either advance individuals towards cultivar release and cultivar release for growing by producers, or we select the best and use them as parents. We make another cross with the best selected parents and create another genetically heterogeneous population. We start the whole process over. So this is, this basic process of creating genetic variation, testing, selecting, and repeating is what drives a lot of the slow changes in a crop species that we see over time as we adapt them to new environments and create new cultivars for new target markets. So a little bit further down in terms of details on this process, most plant breeding programs consists of what we call sort of a breeding pipeline that look like this, that can be represented as sort of a pipeline format here. And you don't have to worry about the numbers inside of this. I'll go through some of the more details here in a second. But what I want you to notice as part of this pipeline is that basically we're starting off with and of course at the bottom of this pipeline we have new varieties that are coming out. But basically this pipeline starts off with a large number of individuals that we're trying to select upon. Okay, we have a large number of candidates for a new cultivar, a large number of candidates for a new breeding parent to start the process over again. So that starts off with a small number, and then as we test more and more, we whittle away and discard the ones we don't want and select the ones that we do want. And so at the same time that we're doing that, as we're whittling away a number of candidates in our program, we're of course also increasing the intensity of our testing. We're testing across more locations, we're testing across more years, we're applying more and more tests for different traits. Maybe it's more disease resistance, more specific quality traits. We increase the intensity of our testing as the number of the candidates in our program decreases, so that the total amount of resources pretty much stays the same. So that's sort of a higher level overview of a typical soybean breeding program. In our soybean breeding program at the University of Minnesota, we, like all breeding programs, start with a cross, and we make maybe 100 or 200 crosses per year. And from that cross, we create inbred populations. We allow those plants, the progenies from that cross, to self-pollinate so that we achieve true breeding inbred individuals. Okay, at the after 3 or 4 generations of self-pollination, and then from those individuals we drive breeding lines. We drive breeding lines, and we start to evaluate them on a breeding line basis. Okay, we start off with single plant rows at the F4-5 stage there at the top, and single short plant rows. And then the individuals that survive that basic visual screening, more or less, go on to numerous yield trials and other trials for testing through the years. And then off to the side of this process, we also have a phase where we do lots of seed multiplication and purification. So we start to get into the later stages of the testing pipeline. We have now candidates that are better candidates for release of a new cultivar. We start to multiply their seeds, and so farmers have enough seeds to grow. And at the same time, we also purify these varieties for the traits that we're interested in, so that the variety is pure and homogeneous for certain desired traits. And then, of course, we test at many locations across our target market, and we also do a fair bit of selection on molecular markers for certain traits, such as disease and insect resistance. Okay, so we have a pretty broad-based program in Minnesota. We select for lots of different traits. We have lots of different markets that we breed cultivars for. And so I sort of categorize these different types of traits into 3 different categories. First of all, we have the production traits. Of course, all parents need to be selected in the program if they have good relative yield compared to the existing check cultivars. They have to have an appropriate maturity for our target zone. And of course, they have to have good lodging resistance. Those are just things that we have to have every year. And also we have our defensive traits, our stress resistance and tolerance types of traits. I'm not going to go through each of these ones in detail here, because I know most of you out there are interested in the end-use quality side of things. But we especially have to have things like soybean cyst nematode resistance and phytophthora root rot resistance, so that we have stable production from year to year in various types of environments that could be affected by these different stresses. And then we have a lot of different quality traits. And the quality trait that we may choose a parent for starting a breeding population could range from, could be yellow hilum, or it could be higher protein, or it could be a black seeded type of variety. And whether we choose a parent for this depends on the target market of the cross. We make a cross with a certain target market in mind. It could be a soy milk market, and if it's a soy milk market, we may want a parent that has the low lipoxygenase trait, for example. And so how we choose a parent for each of these different types of quality traits depends upon the specific target market in mind when we make a breeding cross to start the breeding process. All right, so let's talk a little bit about the tools we use in our program to accomplish what I just described. We have lots of small plot planters and harvesters that are literally able to plant out thousands of breeding lines in a single field. We have various disease and insect screening nurseries, so we breed for good defensive traits, iron deficiency, chlorosis, aphid resistance, phytophthora resistance, and so forth. We have UAVs in the program, actually, that help us take notes on maturity and various disease resistance, such as iron deficiency, chlorosis tolerance. We have near-infrared reflectance technology and good calibrations for allowing us to predict seed composition and seed quality in a high throughput, inexpensive manner. And of course, we have a DNA molecular marker lab that allows us to make selections upon certain traits if we happen to know some underlying genetic markers or the actual genes controlling some of these relatively simply inherited traits. So we use all these types of tools in our program to ultimately drive a new cultivar. So I want to say a little bit more about our near-infrared reflectance spectroscopy calibration capacity. We have a good calibrations. Of course, we can predict protein and oil fairly well, but we also have some good calibrations for carbohydrate composition and fatty acid composition as well. And this allows us to screen hundreds of samples per day in a very inexpensive fashion, so we can use these predictions, these composition predictions from the NIR throughout the whole breeding process to make sure that the things coming out at the end of the breeding pipeline meet the quality targets that we're desiring. So with that background here, I just want to cover a few examples here of some things that we're doing in our current breeding program. I want to talk a little bit about our food type soybeans. I want to talk a little bit about high oleic fatty acid soybeans. I want to talk a little bit about modified carbohydrate profile soybeans and just touch on some things related to low allergens in soybeans. Thanks to the efforts of my predecessor, Dr. Jim Orff here at the University of Minnesota, we have a healthy food type breeding program where we breed soybeans of all different shapes and sizes and compositions to meet the various markets at the bottom of this slide. So just an example, I can't go through all those, of course. So just an example of what we do for breeding new natto cultivars. So for natto cultivars, we basically select for these things listed here in this slide. Of course, we want very small seed size, a good clear hyla, and round seeds. We also want to make sure that the natto seeds have good water uptake. There's a very low stone bean, or actually no stone beans is desired, and we don't want any absence of seed co-cracking. So these are the types of traits that we would screen in a natto cultivar type for a natto cultivar target market. All right, so what about soybean meal here? So just thinking about soybean meal and the limiting factors in that and how we can improve that. There are a lot of, soybeans are, of course, a great protein source with a great amino acid profile. But there are a lot of things in soybeans that reduce their value as a meal. A lot of these things can be deactivated with heat treatment. But of course, heat treatment also lowers some amino acid digestibility and also the overall energy value. And so there are a few things here that we can modify genetically soybean seed to increase the value of the soybean meal. So of course, the feed value goes down because of all these different factors. And what I want to concentrate on here are the presence of oligosaccharides, as well as some anti-nutritional factors such as protease inhibitors, lectins, and some allergens. So starting off with oligosaccharides, oligosaccharides are a form of carbohydrate in the seed. Basically, those come in the form of what are raffinose, the compounds raffinose and stachyose. And the deal with these compounds is that these are carbohydrates that are not really available to monogastric animals, energetically. And worse yet, these are actually fermented in the lower gut, causing flatulence and basically overall reduced feed efficiency. And so they lower the energy value of the feed, right? And so there are genes out there that there are variants of genes that act in this raffinose and stachylose biosynthetic pathway. And there are variants of genes that reduce overall raffinose and stachylose and increase overall sucrose, which is a more available form of carbohydrate. And so we've been working with these genes in the program, the RS2 and RS3 genes, and we have a couple of those and a lot of breeding lines. Here are just a couple of examples of breeding lines with RS2, a variant of RS2 that lowers the oligosaccharides. And you can see that for these two here, we have greatly reduced raffinose and stachylose and slightly increased sucrose. We're also working on a variant of RS2 with a variant of RS3 to increase sucrose further. So on the allergen and anti-nutritional factor side, we've been working with these things that have been called the triple null soybeans, which have been developed by Monica Schmidt, Elliot Herman, and Ted Heimowitz in a Williams 82 background, which is a maturity group 4 type of soybean variety. They bred the soybean variety, and they showed that it has comparable amino acid profile to the normal type of soybean, but nicely it also has basically no lectin, very, very reduced level of Hunus trypsin inhibitor, and a reduced level of the P34 allergen. And so we've been working with this, and we bred this now into elite soybean varieties adapted to Minnesota, and we hope that there could be some. We hope that there could be some application, some use of this in feed, as well as perhaps some whole soybean products such as tofu or soy milk. All right, and then a little bit on high oleic here. We have a non-GMO form of high oleic soybeans where we're using some genes discovered at the University of Missouri to greatly increase the oleic acid in soybeans to make the fatty acid profile of these soybeans look more like olive oil than they do normal soybean oil. So you can see that we have some breeding lines in the program here that are high oleic, designated by the HOLL in their name down there, which are highlighted. And if you look way to the right side of that table, you can see that their oleic level is nearly 4 times, in one case there, than that of the Czech. And they also have good yield. And so we're pretty confident that we can drive good high oleic soybeans that have comparable yield to existing cultivars out there. So we're excited about that. Let's talk a little bit about some future pipeline potential here in our last few minutes. And to do this, I want to just motivate this topic through the discussion of some technologies and compare them to what I just talked about. And so we covered basically, what we do in our basic breeding program using sexual recombination and selection. I now want to talk about mutagenesis, which is an old technology, but is sort of new again in some ways with applications, with advanced applications of molecular markers and genome sequencing, as well as genome editing. And at this point, I want to acknowledge a colleague of mine here at the University of Minnesota, Dr. Bob Stupar, who specializes in the area of functional genomics and the application of technologies like this to cultivar development. So imagine you want to develop a new cultivar with a completely novel trait. And there's no variation for that trait out there within your elite breeding germplasm contained within your breeding program. So what you might do is you may go to the germplasm collection, say, for example, the USDA germplasm collection. You may search that germplasm collection, which is very diverse in terms of its plant types and seed composition and so forth, for the trait that you're interested in. Look for variation. Right? And that's basically what was done in those examples I just talked about a few minutes ago. This is exactly what was done in the case of the non-GMO high oleic soybeans. Somebody went to the germplasm collection and they searched as many accessions as they could, and they eventually found those few soybean accessions that had elevated levels of oleic acid, and then found the genes related to those and bred those into elite germplasm. That was done by Kristen Ballou and Grover Shannon at the University of Missouri. But what happens if you go to the germplasm collection and you're looking for variations for your traits and you come up empty-handed? You can't find anything. Okay, so what can you do in this case? All right, so one thing you can possibly do is you can embark upon a process called mutagenesis. And basically mutagenesis just refers to a change in DNA sequence that is stably inherited. And when you use mutagenesis in the context of cultivar development, usually you induce the mutations in a laboratory setting to increase their frequency. And so I guess I want you to notice that, when we talk about a mutation, we just talk about a mutation being a change in DNA sequence. And ideally, this change in DNA sequence would result in some simple change in plant phenotype. All right. So this isn't to say that this is something scary or creates something freaky. All right. So that's not really what we're talking about with the mutation. Mutation just means a change in DNA sequence that results in a change in plant phenotype. And these can occur in nature, they can occur in a laboratory, or they can be targeted, like I'll talk about in a minute here. So here's an example of a mutation population created here by some of my colleagues here in Minnesota. So what they did is they took a lot of soybean seeds and they applied Fast-Neutrons to these soybean seeds. They grew them up in the field, and they looked for soybeans that had different phenotypic characteristics that they were interested in. And then they studied the genetics of those characteristics. And we are now currently applying some of those findings from this mutation population to applied cultivar development. So here's an example of something that came out of that population, out of that Fast-Neutron mutation population. And this is an example of a new soybean, I'll call it a new soybean genotype that has very, very, very elevated levels of sucrose. The soybean variety has about twice as much sucrose as a normal soybean variety, okay? And it has about half the oil content. You see that the seeds actually look visibly different, being a little more shrunken than a normal soybean seed. And then what my colleague Bob Stupar has done since then is he's used this change in this mutation that causes change in phenotype, and then eventually knocked out the gene underlying this phenotype to create another version of this trait. All right, so that's a little bit about mutation. Let me talk a little bit about gene editing. And I'm sure a lot of you have heard about gene editing before in the popular press and so forth. But when we refer to gene editing, also sometimes called genome editing, we're basically referring to a set of site-directed DNA sequence modifications. All right, so the key phrase here is site-directed. So under mutagenesis, under a natural breeding setting, we're relying on natural random mutations to create the variation that we want. Here we're going to be directly inducing mutations at specific sites. So another trend I want to talk about briefly is these rapidly declining genotyping costs. And this has largely been driven by changes in advancements in DNA sequencing technology. Basically, in my own lab, this is my own experience here. 1 year ago, I paid, roughly about $3 per sample to run only 2 SNP markers to select upon some traits we're interested in. And now through working with some vendors that are applying some more advanced technologies, we're able to get that down to $1.50 per sample for 25 SNP markers in this case. And so we can select upon many, many traits with all these different SNP markers. So why does this matter, you might think? Okay, well, so the combination of gene editing and cheap molecular markers gives us the tools to basically that are inexpensive enough now to allow us to stack together many different traits on top of themselves. All right, so I would say that these tools, gene editing, mutagenesis, and of course natural variation combined with molecular markers, allows us to stack multiple traits together, and this is now more feasible than ever before. So let's just take an example here. So, I talked about a lot of these traits in my presentation. So let's say that you want a new soybean variety with high oleic fatty acid. Perhaps you want a modified carbohydrate composition. You want Mutagenesis-free soybeans for some reason, for some soy milk application, and you want them to P34 null. To get this type of soybean variety, you basically need to alter 8 different genes. And this is possible with traditional breeding, but it's very, very difficult. With gene editing and molecular markers, this is much more feasible than it ever has been before. So I think this is going to be basically the wave of the future, is trying to bring together all these different traits into a single cultivar and also good agronomic performance. So I guess I want to end by saying that we have a lot of different ideas on the soybean breeding side. We have a lot of different technologies and tools at our disposal. We don't always have a very good finger on the pulse of what is desired in the marketplace. And so what I want to emphasize is that there's good communication between end users and food manufacturers and the soybean breeders. You may have thought that a certain type of seed composition wasn't possible before, but the tools and the technologies have advanced so much that it could be very well possible now. So reach out to us and help us put our finger on that pulse of what is desired out there in the marketplace. So with that, I'll just end with these final thoughts. Basically the time is ripe for these types of advancements, and I think communication between breeders, geneticists, and end users and manufacturers is key to make all this happen. So With that, I would like to thank you, and I look forward to interacting with you at the panel session.